Okay, so today's speaker is Boll from the Trend Lab. I actually don't know what he's talking about. He's going to be talking about rethinking the barrier, how gram-negative bacteria remodel their outer membrane to increase fitness.
Hi, everyone. Hey. Okay. So today I'm going to tell you about how a gram-negative bacteria actually modify their outer membrane to promote survival in different environments. So I'm in the Trent Lab, and we really study gram-negative bacteria. And this is just a cryo-electron micrograph of an E. coli cell. And all gram-negatives have these two membranes in the cell envelope. The inner membrane, which is like most biological membranes, is composed of glycerophospholipid. There's a thin peptidoglycan cell wall inside the cell envelope of these gram-negatives. Additionally, gram-negative bacteria have this asymmetric outer membrane, where the inner monolayer is made up of phospholipids, but the outer monolayer on the surface of the cell is made up of this molecule called LPS, or lipopolysaccharide. Lipopolysaccharide is composed of three different domains. The membrane anchor lipid A domain. There's a core oligosaccharide domain that connects the lipid A domain to this repeat carbohydrate called O antigen. While most gram-negatives produce LPS, many mucosal pathogens actually produce a modified form of LPS that lacks this O antigen, and those that component is actually called lipo-oligosaccharide instead of lipopolysaccharide. That'll be important for the talk today. LPS, and specifically the lipid A domain of LPS, is important in biology because lipid A is one of the most potent stimulators of toll-like receptor 4, which is found on all mammalian cells. And once lipid A is bound by TLR4, it activates immune pathways that can produce a very powerful immune response. Lipid A is synthesized via 9 enzymatic steps. These are constitutively active, and this is one of the most highly conserved pathways in gram-negative bacteria. So this is prevalent in most gram-negatives. The first 7 enzymatic steps of this pathway are absolutely essential for survival of gram-negative bacteria. These last 2 enzymatic steps, the LPXL and LPXN step, are responsible for acyl chain addition, and these vary a little bit between gram-negative organisms. This is a student in the lab a couple years ago, Tommy. He loved lipid A so much that he decided to tattoo it on the back of his leg. This is the final product that's made by most gram-negatives. This is the canonical lipid A structure. It's a disaccharide that's bisphosphorylated, which give it a negative charge, and it's acylated. And this is the structure that is made by most gram-negative pathogens, or by most gram-negative bacteria. Gram-negative pathogens have actually evolved enzymes to modify this structure and produce different lipid A structures. And so take, for example, here, Salmonella can actually add an aminoarabinose compound or a phosphoroethanolamine. It can add and remove different acyl chains. And by adding these different modifications to lipid A, it changes the ability of TLR4 to detect these molecules. So it actually can camouflage these pathogens from the immune system so that they can cause infections. In addition to evading the immune system, these different modifications, such as sticking a phosphoethanolamine onto this phosphate group, actually neutralizes the negative charge of the molecule. This is important for antimicrobial peptide resistance, which is what I'm going to focus on today. So a lot of antimicrobial peptides that are made in your body are made by other bacteria in nature are ingredients in Neosporin, the topical ointment that you put whenever you get a cut, prevents gram-negative infections. Colistin is used in the clinic. It's actually the last resort antimicrobial that's used to treat gram-negative bacterial infections. So when all other antibiotics fail, they then treat patients with colistin. And over the past decade or two, many bacteria have evolved mechanisms of drug resistance, and we have lots of multi-drug resistant bacteria these days. So colistin has been used more and more over the past couple decades.
Quick question. What does the cyclic nature of the peptides give? Like, why is that critical to the antimicrobial nature?
So, I don't know. I'm not sure about the cyclic nature. I know that cyclic peptides are, it's what drug companies want, or cyclic peptides. I think it's because they probably enter the cell, or more easily enter the cell, but I'm not exactly sure what the cyclic nature is. The important part of the lipid tails interact with the membrane to form pores, I know. But yeah, the cyclic nature, I'm not exactly sure. But yeah, so colistin use has been, it's been more widely used over the past decade or two. And because it's been more widely used, resistance mechanisms have emerged. So I'm going to focus on a bacterium today called Acinetobacter baumannii. Acinetobacter is found in the US, mainly in hospitals and other in clinics. It's really not found in the environment. Acinetobacter was not found in the US up until the Iraq War, when lots of soldiers that have been wounded actually brought this back, and it's now found throughout the US in most healthcare facilities. Acinetobacter tends to, it causes a wide range of diseases in people. Infections manifest as pneumonias. They can go septic and cause lots of problems in people. Acinetobacter is unique in its ability to acquire drug resistance, and it can actually survive desiccative environments for months and years. So it can survive on inanimate objects in these hospitals for up to years. So it's becoming it's an emergent pathogen that's becoming a problem in the clinic. What I told you about was CAMPs can interact with lipid A, these hexaacylated lipid A molecules, and they can perturb the membrane to lyse the cells. But when I came into Stephen's lab, we started looking at the lipid A biosynthesis pathway in Acinetobacter. And what we found is that Acinetobacter actually hyperacylates its lipid A molecule. So it adds an extra acyl chain to its surface exposed lipids, and that actually works to prevent these CAMPs from lysing these cells, the increased acylation. So what we found was that the canonical lipid A in gram-negatives, the LPX enzyme in Acinetobacter actually adds 2 separate acyl chains to this molecule. And this is the first example of a dual acyl transferase, the first time that one has been described. And so this is just a little bit of data from that paper. When we knock out the gene that encodes the LPX enzyme, these cells no longer produce the hyperacylated lipid A, and they're more susceptible to killing by polymyxin B and colistin. And this is just a viable count of cells with increasing concentrations of these polymyxins. Additionally, we did a desiccation assay where we took a number of our strains, including the LPXN mutant, and what we did for these assays was we stuck these cells, an equal number of cells, on polystyrene for 48 hours. We then resuspended the cells in LB and looked to see which cells were still viable or could still grow. And as you can see, after desiccation, the LPXN could not grow, or we could not recover any viable colonies, suggesting that this hyperisolation in lipid A protects from antimicrobial peptides and desiccation. But when I started working on Acinetobacter, I was really interested in this paper that was published in 2010, and because it really challenged what we think about the importance of lipopolysaccharide in gram-negative bacteria. This paper showed that when they played Acinetobacter baumannii cells on colistin, they could then isolate colonies that no longer produced lipopolysaccharide, which was thought to be essential for gram-negative cell survival. In these LOS or LPS or LOS deficient cells, they still produce both of these membranes. It's really hard to see in these pictures, but they do still produce these two membranes that's indicative of a gram-negative. So we wanted to, one, confirm that these cells actually did not have lipopolysaccharide, and ask the question, how did these cells actually survive without LPS, whereas other gram-negative bacteria seem to be dependent on LPS for survival. So what I did was I took our Acinetobacter strain, we plated it on colistin, and we isolated what we thought were LPS or LOS deficient Acinetobacter strains, and then we replicated on vancomycin and colistin. So our wild-type strain was sensitive to colistin, and once we isolated LOS deficient colonies, which we verified using a number of techniques, we found that there was a 500-fold increase in the NIC of cells that lacked completely shut down LOS biosynthesis. Interestingly, though, Acinetobacter is highly resistant to vancomycin. All gram-negatives are highly resistant to vancomycin. Vancomycin is used as a last resort drug to treat gram-positive infections. But it's not useful against gram-negatives because they have this outer membrane, and the vancomycin cannot actually enter the cell to kill the cell. So whereas Acinetobacter was highly resistant to vancomycin, once it shut down LOS biosynthesis, it became very sensitive to vancomycin.
So actually, I didn't think about this when I saw it the last time. So does that mean that possibly some of these colistin-resistant strains could be treated with vancomycin?
Yeah, definitely.
Has anybody tried this, to your knowledge?
No.
Because they wouldn't do it in the hospital.
Yeah, no, this is
right?
the first report of this, that it's actually it sensitizes it to vancomycin. Vancomycin is used widely in the hospital, so it could definitely be a treatment.
But what I mean is, normally a doctor would not apply this because they'd say this is just useless.
Yeah,
right?
Exactly.
And it might actually be helpful.
Yeah, exactly. And we also see that antimicrobial resistance to a whole number of different antimicrobials changes when these cells lose.
So have you somehow communicated that to the people that might make those decisions?
Yeah, I mean, it's published, so, and
that's
not the same thing.
Yeah, no, I mean, no, I guess we haven't. How would I go about doing that?
I mean, you have like reports of these colistin resistance in the clinic. I mean, you could in theory contact physicians so that they would have something.
Yeah, I mean, we probably should. We're working with an ID doc that's given us a bunch of clinical strains, and yeah, he hasn't said anything like that, but I'm sure that I could talk with him and we could figure something out.
So it'd be worth passing that result back again, because that's a.
Yeah, I mean, they're not going to read the literature and find it, right? I mean, even the first time I saw it, didn't click, right?
Yeah, there's a couple of different antibiotics that we found that this thing becomes much more sensitive to after it uses LOS. So
I think ultimately there would have to be case reports in the clinical literature that say, oh, we gave this drug.
Patients reported that, even colistin were resistant to vancomycin.
Yeah, or
Exactly.
you could synergize drugs. You could give them both colistin and vancomycin. They should take care of both, right? I mean, gram-positives, gram-negatives, everything. Yeah, I know.
Do you know if they can re-evolve or colistin resistance?
They do, yeah, and I'll talk about that at the very end. But yeah, in vitro definitely, especially if you can select for them on the LOS deficient colonies on vancomycin, you can get colonies back that have reverted and are now producing LOS.
So is this the only method the resistance is applied?
No, there's other, so it can, this is the only mechanism of high level resistance. So clinical resistance to colistin is defined as having an MIC of above 4 micrograms per mil. This level of resistance gives you about 30 to 1,000 micrograms per mil. The other resistance mechanisms that have been characterized in the literature, it's somewhere between like 4 and 8. So it's not.
Did they change the number?
They modify the lipids on the outer membrane. Yeah,
But it doesn't induce the vancomycin.
no, they're not sensitive to vancomycin. Yeah.
So is that like a sweet spot where the bugs will kill you and you can't do anything, or is it a resistance level where the colistin still does something?
Yeah. So colistin is still, So
Sweet
colistin,
spot for the
Sour spot.
the highest what they treat, they can give colistin up to 8 micrograms per mil, and it might be too toxic too.
Okay. But in general, it's not good to take colistin, right?
Yeah, no, it's pretty toxic. That's why it's the last resort option. It's just there's much better drugs, much less toxic drugs that you can use to kill it. So, okay, so in this strain that I'm showing you right here, these LOS deficient mutants that I'm showing you, we did whole genome sequencing and we found that the mutation was in the LPX-C enzyme, this is acetyltransferase, and when we constitutively expressed LPX-C back in trans, it restored the wild type phenotype. And there were no other secondary mutations in these strains that we sequenced. So this is just knocking out the lipid biosynthesis pathway. Okay.
So did you sequence several isolates?
Yeah. Yeah, I've sequenced probably 20 of them. I mean, there are a couple of secondary mutations in some of them, but it's nothing consistent.
Sorry, it's the same gene or it's always the exact same?
So it's one of 3 genes. Yeah, so it's one of the 3 first steps, LPX-A, C, or D, in that lipid biosynthesis pathway. That's
Should have a mutation.
Yeah. It's a variety of different mutations, yeah. So we collaborated with an infectious disease doc, and he gave us a number of recent clinical isolates of these Acinetobacter strains, and we, so the previous paper that we found in the literature had only examined this 19606 strain, and that's what the data that I just showed you, that was what that was from, that we were able to verify what they had done. So he gave us a number of strains, clinical isolates, all isolated from different sources. And we wanted to ask the question, can all Acinetobacter strains survive without LOS or LPS? And so what I did was I just did our screen that I showed you. I plated all of these on colistin, and we just looked to see if we could isolate LOS deficient colonies. And as you can see, in about half the strains, we could isolate LOS deficient colonies, and the level of resistance varied from 30 to about 1,000 micrograms per mil. There were some strains indicated by this NA that we could never isolate any LOS deficient colonies using this this method or this screen. So we inferred that these Avamani isolates could not survive without LOS. And so this really gave us a good model system to study the essential components for survival without LOS.
So they were not to begin with LOS minus?
No. So we're just plating the wild type on colistin, and then the colonies that pop up, we're just checking to see if those don't have LOS. And about 90% of them don't. whenever we get colonies back. But in these strains, such as 17978, we could never isolate any LOS deficient colonies. So to really, so one of the approaches that we took to understand how these cells are surviving without LOS was we did a saturating transposon screen using TNC in 17978. This is the strain that cannot lose lipid A or LOS. We took our library and selected for growth on high levels of collistin, and we got colonies back. We verified that the 17978 strain with these transposon insertions did not have LOS. And all the transposon insertions that we were then able to sequence, the transposon insertions, and all the insertions were in the gene encoding this protein right here, this penicillin binding protein 1A. PBP1A is involved in peptidoglycan biosynthesis or cell wall biosynthesis. And it's a multi-domain protein. It has two enzymatic activities. One is a transglycosylase activity, and it has a transpeptidase activity. So this is just a short summary of peptidoglycan biosynthesis. I'm not going to go through this. All you need to know is that the transglycosylase activity extends these chains of peptidoglycan, whereas the transpeptidase activity forms peptide bonds before these glycan chains to form this, this meshwork that is the peptidoglycan cell wall. And this holds the cell, it really gives the cell its shape, and it holds these bacterial cells together. Okay, so just to verify that these 17978 transposon mutants did not have LOS, what we did was I took either a wild-type strain or LOS-deficient mutant, and we grew these in the presence of radiolabeled P32. The P32 gets incorporated into the lipid A at the phosphate sites. We then isolate the lipid A, and we run it on a TLC plate and separate these lipids. And as you see in our wild-type strain, this is the lipid A banding pattern that we see. In the PBP1A mutant, the lipid A does not change. But then when we select for colonies without LOS on collistin, those colonies that we've selected for no longer produce lipid A. Additionally, we tested to see if these LOS deficient cells activated TLR4. So we have a tissue culture cell line that expresses the human TLR4 receptor, and it's linked up to a reporter, and so we can measure TLR4 activation using a calorimetric assay in the spectrophotometer. So as you see, our wild-type strain highly activated TLR4. This is increasing the number of cells that we added. This is just TLR4 activation. Our wild-type strain activated TLR4, whereas our LOS-deficient strain, which is this red line, did not. And it activated it about as well as a gram-positive Staph aureus, which does not produce lipid A or LOS.
What's the PBP control?
So PBP1A, it just it aids in peptidoglycan biosynthesis and maintenance. So it just works to extend these glycan chains and to link the glycan chains.
So, yeah, I guess I can start understanding control. Like, is that control for?
What is that?
Oh, so why do you have that? Why did you use that?
Oh, so we did. So we wanted to know what factors were important to allow these bacteria to survive without LOS.
Oh.
And so by knocking out this PBP1A protein, it then allows these Acinetobacter cells to survive without LOS. Yeah. Okay. So these 17978 strain that had PBP1A knocked out, it no longer produced lipid A or LOS, and it did not activate TLR4, indicating that it doesn't produce lipid A. So how does PBP1A fit into all this? So I've told you about two Acinetobacter strains so far. 17978, which does not activate, or which we cannot isolate LOS deficient cells without knocking out this PBP1A protein. In 19606, which when we play marcolistin, we get lots of LOS deficient mutants. So we looked at the mRNA concentration of PBP1A in these two wild-type cells. In 17978, it seems to produce a lot more of this PBP1A transcript, about 80-fold more than the 19606 strain. And if we look at the proteins, 17978 produces PBP1A, but we could not detect it in the 19606 strain, suggesting that low levels of PBP1A in 19606 is required to be able to select for these LOS deficient bacteria. We then did some genetic analysis just to confirm. So we looked at there's a catalytic residue in the transglycosylase domain of PBP1A and a catalytic residue in the transpeptidase domain of PBP1A. And so we wanted to look and see if these, the enzymatic activity of these domains was important for the survival of these cells without LOS. So in 19606, we can isolate LOS deficient colonies, but when we add PBP1A back in on the plasmid, we lose this ability to isolate LOS deficient colonies. When we knock out the transglycosylase activity, we can recover the LOS deficient colonies. In the transpeptidase, when we just knock out the transpeptidase activity, we can no longer isolate LOS deficient colonies. So this transglycosylase function is required to block the loss of LOS. Additionally, in 17978, we could never, in our wild-type strain, we couldn't select for LOS deficient colonies. When we knock out PBP1A, we get lots of LOS deficient colonies. And the transglycosylase activity is required for LOS deficiency, as you can see here. So this really just suggested that this transglycosylase activity of this protein is what is inhibiting the 17978 strain from surviving without LOS. So to really understand how this is all working, what we needed to do was look at the total peptidoglycan composition in these Acinetobacter cells, because we really wanted to understand how PBP1A was changing the cells to allow them to survive without LOS. And so we collaborated with Waldemar Vollmer. He's at Newcastle University in Great Britain. And we isolated total peptidoglycan from these cells. And we looked at how long the glycan chains were, how cross-linked they were, lots of different things, lots of different numbers on the slide. But there was really no differences in our wild type and our PBP1A mutant or the LOS deficient bacteria.
So sorry, I don't see that.
Yeah, so if average chain length didn't change, so 30.47 in the LOS mutant, it was 34 in the.
So numbers are the same across rows.
Yeah, everything was pretty much the same. I mean, it was within the variation.
It's a general rule I always tell everybody never to put tables like that on the side.
Yeah, no, I totally agree with you because the data is just overwhelming and you can't make anything of it. But there's really nothing to make of all this. It's all pretty much the same. So yeah, at this point we didn't really know what to do next. We were wondering, okay, how is this changing the cell to allow it to survive without LOS? And we were somewhat frustrated at this point, but we just decided to go ahead and look at some transcriptomics from these various cells and take a more global approach of what's going on in the cell. And so we did some RNA-seq on these mutants, the PBP1A mutant and the LOS deficient mutant. And we just looked at what pathways were changed. We did pathway analysis using the transcriptomics. We looked to see what pathways were upregulated and what pathways were downregulated. And what we found is that Boll lipoprotein transport was highly upregulated in both the PBP1A mutant and the LOS deficient strain. And it was conserved among these two strains. Low lipoprotein transport one of the big problems in gram-negative bacteria is everything in the cell is made at the inner membrane, but it has to have its own set of unique transporters to get it from the inner membrane to the outer membrane. And there's no energy in the periplasm, so it has these dedicated systems to move things from the inner membrane to the outer membrane. And this system is involved in moving lipoproteins from the inner membrane to the outer membrane. Additionally, phenylalanine metabolism, this pathway was conserved among these two mutants in our downregulated genes, but I'm not going to talk about this today. So this really suggested that low lipoprotein transport was important. And we confirmed this using qRT-PCR, where we were looking at expression of this Boll A gene. This is the periplasmic chaperone that actually binds to the lipoproteins and moves them from the inner membrane to the outer membrane. And as you can see in our PPP1A mutant, there's about a 10-fold increase of Boll A transcription. In our LOS mutant, there's about a 100-fold. In the Boll-type 19606 strain, it's about a 10-fold increase from 17978. In the LOS mutant, there's about a 100-fold increase. So we know that lipoprotein transport and movement of lipoproteins from the inner membrane to the outer membrane is upregulated in these PPP1A mutants and the LOS deficient strains. So that suggested that lipoproteins are important for survival without LOS. And so we just wanted to go ahead and take a global look at the global transcriptome of multiple Acetobacter strains. And so what we did was we took these 4 strains, these are clinical isolates, Amoii isolates, and we made all of these LOS deficient, and we just did whole transcriptome sequencing, compared the genes back to the wild type, looked for upregulated pathways and downregulated pathways, and among these 4 strains, we found that 5 pathways were upregulated and no pathways were downregulated among all of these strains. And the 5 pathways that were upregulated were lipoprotein transporters, putative lipoproteins, these multi-drug efflux pumps, a two-component system, and phospholipid transporters. But the 2 pathways with the most, with the biggest changes in gene expression were our lipoprotein transport and these putative lipoproteins. So this really suggested that maybe in the absence of these cells producing LOS, maybe these lipoproteins are somehow being redirected onto the surface of the cell to permit these cells to survive without LOS. And so what we wanted to see was, are these putative lipoproteins actually found on the surface of these bacterial cells? And to really answer this question, we use this molecule right here. It's called sulfo-NHS-LC-LC biotin. It's impermeable to the outer membrane of these gram-negative bacteria. What it does is it reacts with primary amines on proteins, and it adds a biotin molecule onto these proteins. So it only biotinylates the proteins that are found on the very surface of the cell. It's been used in E. coli, in numerous other systems, Vibrio, just to show the surface permeability of these bacteria. And so what we do here is we take our strain, our wild-type strain or our LOS deficient strain, and we just add in this molecule in with our strain. It biotinylates all the surface proteins. We then wash away the sulfo-NHS-LC-LC biotin, and then we fractionate these cells. So we have a soluble fraction and a membrane fraction, and then we run them out on an SDS-page gel, and we probe the gel with an antibiotin antibody, so then we can see which proteins actually get biotinylated. As you can see, none of the soluble proteins are biotinylated, suggesting that this molecule does not enter the cell. It's impermeable to the outer membrane. Only membrane proteins are biotinylated. As you can see, there's a number of proteins in our wild-type fraction, membrane fraction, suggesting that there are proteins on the surface of Acetobacter. When we looked at the LOS deficient strain, the protein labeling was very different. Specifically, there were these proteins that were pretty prevalent at these lower molecular weights. And through further characterization, we found that these 2 bands right here in this red box actually were 3 different proteins, 2739, 1944, and 1945. And you see from our transcriptomics, 2739 was our most upregulated protein. 1944 and 1945 were also fairly upregulated. So what we took from this was that LOS deficient bacteria were actually increasing lipoproteins on the surface of these bacterial cells. And we think that somehow that compensates for and allows the bacterium to survive without LOS. So this is our model. In wild-type cells, they produce LOS in their outer membrane, but in the LOS-deficient Acinetobacter, we think there's probably a mixture of phospholipids and there's many surface-exposed lipoproteins. So I've been working on this a little bit more, and what we have also found is that glycosylation actually seems to be important for survival of cells without.
Oh, so I was wondering, have you measured the concentration of the surface-exposed protein versus the concentration of LOS traditionally found in bacteria?
No, we haven't.
Okay.
No. And I don't know if you can yeah, it'd be a good experiment to do, for sure.
What is the explanation for worker transcription, especially for these babies? Because they seem to be upregulated.
Yeah, they're not expressed in the wild-type cells for whatever reason. We can't detect them.
Explanation why that's happening?
I don't know. I really don't know why they wouldn't be expressed in the wild type versus why they would be expressed in the LOS deficient, unless it's some sort of mechanism that allows these cells to alter their outer membrane when they sense stress.
So you don't know secondary mutations.
So these strains that we're looking at don't have secondary mutations. We've sequenced the genomes and they don't have secondary mutations. So it's totally transcriptional. Is what we're seeing with the increased activation, the increased production of these lipoproteins. So
are they completely not around before, or are they on a different membrane?
So these are whole cell lysates. So these are total composition of the cell, and we can't detect them in the total composition of the cell. So
but
it would
be on the inner membrane, and maybe they get.
But this is total membrane, so even if they're on the inner membrane, we should see them.
But you said this doesn't.
But this only, no. So I get what you're saying. So yeah, it's 2 different things. Sorry. I've also done this where I've just taken lysed cells and added in this compound. So it should, and I still don't find it. Yeah. So.
Do we know what those proteins' functions are when they're not on?
There's no, I even had to predict that there were lipoproteins. Yeah, using
So
solvers.
if you look at the promoters, can you do a reporter assay just to see that the promoters are these strains?
Yeah, we could definitely do that. I mean, we could do that, and we could figure out what regulates them and that. But we haven't done that. So yeah, so we think that these lipoproteins are enriched on the surface of these cells when they don't produce LOS. In addition, since this was published, what we found was that glycosylation, or production of glycans in these bacteria, seems to be essential for survival of LOS deficient A. baumannii. And one of our hypotheses is that these glycans that are produced by the cell are actually added on to these lipoproteins. And that would give them some of the same biophysical properties as LOS, where they have this carbohydrate motif and this membrane, these acyl chains that are found in the membrane. So it could help maintain the integrity of this outer membrane.
Is there anything like a lipid fluidity assay or something like that you could say because of these additional protein? I don't know.
Yeah, I don't know. I haven't looked into it, but it's a good idea.
Do you know the shape of the glycosylation on there? Is it the same as the one on the LOS, or?
So there's only one. Well, so there's 2 glycan biosynthetic pathways in Acinetobacter. I've knocked one out; it has no effect. But there's a second one that does seem to have this effect. And this glycosylation pathway, here, I'll give you this slide, it's my next slide. So this glycosylation pathway that does seem to be essential is responsible for the production of capsule and these glycoproteins. So where I'm at now is trying to determine whether capsule is important or these glycoproteins, or glycosylation of these proteins is actually important. And so this is the data from the glycan pathway analysis. Our wild type, we can get LOS deficient colonies back when we knock out the initiating glycosyltransferase for this pathway. So this is the enzyme that actually starts building this glycan. We cannot recover any LOS mutants. And this, we can complement this. I've knocked out this WZX enzyme. This is responsible for the polymerization of these glycans and for them to be exported as capsule. When I knock out the gene encoding this protein, I can still get LOS deficient mutants. So it suggests that capsule is not the important thing. However, when I knock out the this protein right here, PAGL-L, that transfers the glycans onto the acceptor proteins, it seems to be essential for isolation of these LOS deficient cells. So this suggests that glycoproteins are the important component and not capsule. So, does that make sense? Okay. And then, so what I've been doing over the last couple of weeks is I generated a glycan antibody that recognizes these glycans. So this is the capsule in our wild type cell, and there's a couple of glycoproteins that pop out in our wild type cell. And in the PAGL-C mutant, which is the initiating glycosyltransferase, we don't see that any of these are produced. And I'm going to use this as a tool moving forward. So, work in progress. So something else that I'm interested in this project is this two-component system. Bacteria use these two-component systems to sense and respond to environmental changes. This BAY-SR two-component system has not really been characterized in any bacteria previously. There's been a couple of reports over it, but nothing that significant has stood out. These were both upregulated tenfold in our RNA-seq analysis. And when I knock out the genes encoding this two-component system, I can no longer recover any LOS deficient colonies, suggesting that it's regulating something that's essential for survival of these cells without LOS. And I've done RNA-seq on the BAY-SR mutant, and we found that it regulates expression of some of these lipoproteins, or it putatively regulates, it may regulate expression of some of these lipoproteins. It also could regulate some of these efflux pumps that we've shown. So it could just be a regulatory mechanism and all fit together. So as for the last thing that I'm doing is I'd like to understand how inactivation of lipid A biosynthesis affects virulence. So LOS deficient A baumannii have been isolated directly from colistin-treated patients. And so I want to know if the loss of lipid A or LOS actually affects virulence of these bacteria. So I did a preliminary mouse experiment in which I took wild-type Acinetobacter and re-inoculated them into a mouse intranasally, so it goes directly into the lungs. It's a mouse pneumonia model. So these bacteria get into the lungs at high amounts. After 48 hours, we sacrifice these bacteria, and these mice, and do colony counts in these different organs. So you can see there's a high bacterial load in the lung.
Not really being fair, right? You save the bacteria and
Yes, sacrifice the mouse, save the bacteria. Yeah.
And then sacrifice them as well.
Yeah. Well, they don't live much longer. Yeah.
But first, you let them reproduce, right? I mean,
Yeah, no, they
colony count.
Yeah.
They're like really happy, right?
So we stick in about 10 to the 8 in this experiment, which is a lot of bacteria, and they replicate up to 10 to the 9, about a tenfold increase. And we find that they disseminate. So this is after 48 hours, we sacrifice the mouse and we take out their organs. And there's a high bacterial load in the liver and in the spleen. So these are actually able to get out of the lungs and move through the bloodstream to peripheral tissues. When we did this with the LOS deficient strain, the inoculum's a little lower, but we did find that there were LOS deficient colonies in the lungs after 48 hours, but we did not find that they were able to disseminate.
Why was the inoculum lower for those ones?
So what I have to do is do the mouse experiment. So I inoculate the mouse, and then I have to plate the inoculum that I'm putting into the mouse. So the next day, I don't know if they're equivalent until the next day. Yeah, and I can't go back and redo the experiment. But I've done this twice, and this was the closest count that I got. This suggests that LOS deficient Acinetobacter are not virulent, but do persist in the lung over time. These mice, when you treat them with the LOS deficient bacteria, they don't get sick. These mice get very, very sick, and by 48 hours, they need to be sacrificed because they're not doing well. These mice don't get sick at all. So it suggests that wild type is virulent, but the LOS deficient ones are not. So what I'm interested in asking next are what are the innate immune mechanisms that keep Acinetobacter from disseminating from the lungs into these peripheral tissues when they don't harbor LOS? Can LOS deficient Acinetobacter revert to wild type over time in a host? So can these things, LOS deficient cells, hang out in the lungs long enough that they acquire secondary mutations, start to produce LPS, and then are they able to infect the mouse like wild type? And what factors are important for Acinetobacter to escape the lungs and disseminate into peripheral organs? So I'd like to do some TN-seq on the wild type strain and see what's necessary for it to get out of the lungs, escape the lungs, and to colonize these other organs. So yeah, so big picture. Gram-negative bacteria can survive without LOS, but the mechanism of remodeling is not understood.
So, have you ever tried a combination between LS minus versus LS plus cell?
So in a mouse?
No, but in
Yeah, so the wild type strain outcompetes the LOS.
so there is a fitness defect.
Yeah, oh, yeah, there's a growth defect. I mean, there's definite fitness cost for sure.
None of the clinical isolates have the LOS deficiency, right?
They do, yeah. So they've isolated LOS deficient cells directly from a patient.
And so it still made them sick.
It's not, yeah, I don't know if that made them sick or something else made them sick, or if there's wild type bacteria present too that made them sick. Yeah, yeah. But yeah, and so the mechanism of this outer membrane remodeling after they lose LOS is not understood. We don't know why PPP1A seems to be toxic to LOS deficient cells, but what I have showed you is that we think that these lipoproteins are being redirected, overexpressed and redirected onto the surface of the cell to compensate for the loss of LOS.
This may be a naive question, I don't know much about bacteria, but are there other strains of bacteria that use this sort of secondary mechanism that you described? It's
never been described before in another bacterial system. No. There's only 2 other bacteria that can survive without LOS. Neisseria meningitidis, causative agent of meningitis, and Moraxella causes inner ear infections in kids. They don't have the same level of colistin resistance when LOS is shut off. So this is the only bacterium that does it like this, I think. Nobody's ever explored those other bugs, but there are a couple other ones that can do it. They can survive without LOS. But no, I mean, out of all the other bacterial systems, none of the other ones can survive without LOS, to our knowledge, and people have looked quite extensive. And so the last part of the talk I've talked to you about glycosylation. We know that that's important in this BASR regulatory system, our fitness determinants for LOS deficient survival of these cells that are LOS deficient. And then I'm going to end here acknowledgements. These are the people that worked on this paper with me. Vincent is the ID doc that gave us the strains. Alex is a graduate student in the lab. Katharina, she did a lot of the neuropeptide analysis in Waldemar. She's in Waldemar's lab. Collaborators, the Davies lab has also been a lot of help during this work, and the Broad Belt group. Jane Broadbelt and Dustin did a lot of the mass spec to characterize a lot of these lipids. Yeah, and then the support, of course. Can't do it without the support. So with that, I'll take any other questions that you guys have.
What's the difference between a lab and
I don't know. She's always just called her lab the groups or the Broadbelt group. So that's what I want. Yeah. There's a lot of them. Maybe it's just pure number of people. Okay. Thanks, guys.